Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for a speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Eric Bishop from Cygnus Technologies.
Welcome, and thank you all for taking the time out of your day to participate in this webinar. Today we're going to discuss a case study for bridging of the F550 to the F550-1 CHO host cell protein ELISA at Cygnus Technologies. Hopefully, this can be used as an example of general expectations as well as best practices for a successful transition between two assays. Here's a brief history of Cygnus Technologies. We became involved in host cell protein analytics in 1998 with the publication of a white paper of our idea of how to monitor host cell proteins. In 1999, we launched our first commercial CHO and E. coli host cell protein kits. We spent much of the early 2000s expanding our menu of generic products to cover all of the commonly used expression platforms. And after doing this and performing 2D Western blot for coverage analysis for many years, we realized that it was not a predictive method for how the antibodies would perform in the ELISA. As a result, we launched antibody affinity extraction in 2014. We have since spent a lot of time incorporating AAE and mass spec into host cell protein analytics. First, a little on the actual host cell protein ELISA. On the left is a schematic of how an HCP ELISA works. This method is the gold standard method for process monitoring and release testing for host cell proteins, primarily because it is the one method that can measure nanogram per mil levels of host cell protein in the presence of mg per mil levels of product protein. It does not require a special level of expertise and is easy to transfer across an organization. Unfortunately, the host cell protein ELISA also has some limitations. At the end of the assay, you read the plate and the software reports a result for each sample. The result does not tell you anything about what host cell proteins are in that sample or which host cell proteins the assay is reacting to. Host cell protein ELISAs are not actually measuring nanogram per mil host cell protein. In reality, is an immunological equivalence relative to the standards. Therefore, the results are subject to change, sometimes significantly, with the change of a kit or of critical reagents. And due to the complexity of making broadly reactive host cell protein antibodies, it can take 12 months to generate new reagents and to make a new host cell protein ELISA. So ideally, you don't want to have to change host cell protein reagents, but inevitably the time will come where reagent change cannot be avoided. So now I'd like to discuss reagent changes and how to make these transitions as smooth as possible and how to make sure the new kit or reagents are acceptable for use to monitor your process. So all reagent changes are not equal and as such do not require the same level of characterization to implement. The first type of change are critical reagent changes. This means that an antibody or standard antigen has been fully depleted and requires replacement. This is especially true for the capture and detection antibodies, as immunizing new animals will almost certainly result in different mix of host cell protein population and anti-HCP antibodies. Non-critical reagent changes are typically more minor, such as the purification of an additional antibody from the same plasma or serum. Another would be the new conjugation of a detection antibody. Non-critical reagent changes still require some level of characterization, but on a much smaller level than the critical reagents. So now that we've identified the types of reagent changes, let's look at what we can do to understand the impact of the changes made and the transition to new reagents. There are 3 steps we follow when transitioning to these new reagents. These are orthogonal characterization, assay qualification, and bridging. So we'll start by discussing coverage analysis, and specifically antibody affinity extraction. As I mentioned in the intro, antibody affinity extraction was launched in 2014 as a method to replace 2D Western blot. We have found that regardless of how a Western blot is performed, the severe limitations of the method make it unable to predict how the antibodies will perform in an HCP ELISA. This diagram is an example of how AAE works. Initially, the same antibodies used in the assay are coupled to a solid support. After extensive conditioning and washing, the sample is loaded. When performing AAE for coverage analysis, the sample is always a null or mock transfected harvest sample. Once the sample has been loaded, the column is washed until the background signal is achieved, and then the column is eluted. So at the end of the experiment, there are two samples. The first is the starting material, representing all the HCP that could find its way into your product. The second is the AAE eluate. This is all the HCP that has specifically been removed by the antibodies. Now, for the last few years, the most common detection method has been large format 2D silver stains. However, more recently, many companies are opting for mass spec detection for the coverage analysis. So with the 2D gels, you get the 2D gel images and a percent coverage, but there's not a lot of other information. When using mass spec, the results are a virtual 2D image that's color-coded to reflect the matched spots, a percent coverage, a list of identified HCPs, the molecular weights, and the isoelectric point of each host cell protein. So as I mentioned in the previous slide, that we launched AAE in 2014, and the most common question that I've had in that entire period is, how do we know the HCPs removed from the sample were a specific interaction? And my response is always, well, how do you know that the spots on your 2D Western blot are specific? Because I've never seen anybody show a negative control blot. However, we have done quite a bit of work to demonstrate the specificity of AAE. So here's an example of the specificity testing that we've done for the CHO AAE column. So in the interest of time, we won't go through the whole experimental plan, but in short, we constructed an antibody affinity extraction column using the anti-CHO host cell protein antibodies, and a second column using non-immune goat IgG. We passed the null CHO harvest sample over each column and performed mass spec identification. Here is the data from the antibody affinity extraction specificity testing for our CHO host cell protein antibody. On the top right, you'll see the loading and elution profiles of the 4 column cycles. Please note that the column is washed to background after each load, and that there is a nice elution peak for each cycle. The non-immune goat IgG is on the bottom right. Again, the column is washed to baseline for each cycle. However, there is not an elution peak for the non-immune goat IgG. This is proof that the antibody affinity extraction is specifically removing host cell proteins from the sample, and that the coverage assessment using AAE is superior to other methods. So we've discussed the basic reagent and assay changes and discussed the fundamentals of antibody affinity extraction. So now let's look at a case study. I would like to take you through some of the work we performed at Cygnus when bridging between the F550 and the F550-1 CHO host cell protein ELISAs. The first thing we do to assess the impact of a critical change is dilution linearity testing. And this is usually done for all in-process and drug substance samples for the purification process to be monitored. So first, what is dilution linearity? Let's start by looking at the table on the slide. You will observe that at 1 to 2 and 1 to 4, the dilution corrected result appears to be increasing. However, at 1 to 8 and beyond, the dilution corrected result stays relatively the same for each dilution. This is dilution linearity. It is important for a number of reasons. First, if you can demonstrate good dilution linearity in all in-process and DS samples, it is a very strong indicator that the antibodies in the assay are a good fit for the HCPs and the process samples. If the dilution linearity is poor in all samples, the reagent selected may not be appropriate for use in this assay. And if there's good linearity in the early process samples, but is lost after a particular step in the process, it is very possible that the purification process is most likely enriching host cell protein, and that is a good signal that needs to be investigated. So once we're completed with the dilution linearity, the next step is the coverage analysis. So here is the large format 2D silver stain detection of an antibody affinity extraction using the original F550 antibodies. Again, we don't want to spend too much time here, but I just want to reiterate that the point of doing coverage is to demonstrate that the antibodies are broadly reactive to the host cell proteins that are in your process. And we've shown the specificity of antibody affinity extraction, so you can see here that it has pulled out a lot of host cell proteins, and the images are very similar. So you can already conclude that they are broadly reactive. However, given the limitations of gel-based methods, the coverage still comes out to be 73%. It's very similar data under a different color scheme for the F550-1. Again, you can see that they are broadly reactive, and in this instance, the coverage came out to be 86% with the -1 antibodies. So now let's look at Cygnus technologies and how we utilize AAE with mass spec detection to characterize the F550 and the F550-1 CHO host cell protein antibodies. Here's a quick overview of the process flow. The samples are fractionated by antibody affinity extraction, then sample prep, method development, LC-MS analysis, and finally report writing. Here we present the coverage analysis by antibody affinity extraction with LC-MS detection for the F550 and F550-1 antibodies. The table on the bottom shows that the coverage of the antibodies is almost identical at around 97%. The virtual gel images on the top are generated using the PI and molecular weight of the identified proteins in the pre and post AAE samples for each antibody. This is more useful than the 2D silver stains because it allows for color coding the spots, making it easier to identify if there's a pattern of missed spots. Here you can see the missed spots in red are mostly evenly distributed, indicating there is not a certain type of protein that is not recognized by the antibodies. In addition to assessing the coverage by mass spec, we can also use the same data to see if the antibodies in the 550 and the 550-1 kit recognize the same proteins. In this case, there is a 96% overlap in the HCP recognized by these two antibodies. This is very strong data to demonstrate that the F550-1 antibody is a suitable replacement for the F550 antibody. The next step for changing to the new assay is qualification in the new assay. This is assessing the ability of the assay to monitor a designated purification process. So we've already discussed the importance of dilution linearity, but the remainder of the qualification will assess the accuracy to demonstrate that the assay is measuring all of the HCP in the sample, precision to ensure that the assay is under control, and range to establish the analytical range of this assay. Additional testing can be performed at this point, but this is a minimum that's required for early stages or early phases. So now let's look at how we perform bridging between the F550 and the F550-1 CHO kits. Bridging is performed by testing representative samples from the purification process in both assays at the same time. Notice that I've highlighted at the same time, as it can be very challenging to compare samples that have been run months or even years apart, especially if they were not stored properly. Bridging will set the expectations of differences and similarities between the two host cell protein ELISAs. I'd also like to take this opportunity to point out that even if the exact same antigen is used to immunize two herds of goats, and the same affinity column is used for purification, there will be differences. In the case of our commercial kits, most companies see very little difference. However, it is not uncommon for HCP results to increase or decrease significantly, sometimes even within the same purification process. In other words, within one purification, some samples may be higher with the new assay, and some samples can be lower than they were in the previous assay. Also, as you've seen in the previous slide, the anti-CHO host cell protein antibodies from the 550 and the 550-1 kits recognize 96% of the same host cell proteins. However, the affinity of the respective polyclonal antibody to certain HCPs might differ, and this is another potential reason for seeing different HCP values in the ELISA. This is one of the major reasons that we try to avoid reagent changes when at all possible. Here's an example of the bridging performed at Cygnus for newer lots of the CHO antibodies. I wanted to show this for two reasons. First, you can see that there will be changes in sample results when changing reagents. Second, to demonstrate the strategy for bridging assays or reagents at Cygnus. For our assays, we maintain a sample library that we can use anytime we're changing reagents to assess the impact to customer samples prior to launching a new kit or set of reagents. Having a sample set in your own labs can help you if you need to perform bridging studies. Also, having the sample library can allow you to trend how the assay is performing over time. Here are some best practices to help ease transitions between two new kits or a new kit. First, maintain a set of controls that are run on every assay performed. Having these controls allows for trending and performance of the assay over time and can be valuable in bridging and also picking up changes in assay performance. Establish a sample library to make reagent changes easier to manage. This also allows for trending of how different assays or reagents have performed over time. If you cannot establish a sample library, then begin collecting representative samples as soon as you become aware that a change is coming. This will allow for timely transition and helps to ensure that you are ready before exhausting the current reagents. Managing expectations. I want to reiterate a couple of points before closing this presentation. You should try to have sufficient reagents to reduce the number of times that critical reagents have to be made. However, eventually reagents will be exhausted and transitions have to be made, so prepare early. Second, you should expect to see differences when changing to a new assay or set of critical reagents. The results can be significantly higher or lower, and that is totally normal. Also, if the result is lower, that does not mean that the new assay is not as good. Just as if the results are higher does not mean the assay is better. It simply means that the goats made a different array of antibodies that react with the HCP in your samples differently than the previous assay. Also, if you have been using the F550 kit for lot release testing, determine what, if any, effect differences in controls and sample values have on your release criteria and document those changes. Changing the immunoreagents changes the originally validated specificity of the HCP ELISA for each product, regardless of whether the measured ppm levels are the same or not. That requires revalidation of the immunoreactivity and updating the HCP ELISA SOP before the new ELISA can be used. In this case, the F550-1, or if you have a process-specific ELISA that can be used for lot release testing of DS. Okay, at this time, I'd like to finish up and use any time we have remaining to entertain a few questions.
Okay, great. Before we get to your questions, we do have some questions for the audience. So if you can use your mouse to go ahead and select the answer that is best for you on your screen. The two questions are, does your company perform antibody coverage analysis internally? Yes or no? And does your company use LC-MS as an orthogonal method to characterize HCPs? Yes or no? Again, if you can go ahead and use your mouse to select which answer is best for you, that will help our speaker. Okay. And then as far as the questions from the audience, we do have a few. Please go ahead and type those questions in when you're done with the survey. But the first question is, how do you identify the HCP molecules that cannot be recognized by anti-HCP antibodies?
So the only really good way to identify host cell proteins that were missed by the antibodies is by doing antibody affinity extraction and mass spec. And when you do that, you can see all the antibodies that were specifically pulled, and you can measure or identify those host cell proteins, identify all the host cell proteins in the pre-AAE sample, and that will give you an exact layout of the host cell proteins that you were able to recognize and also the ones that you miss.
Okay. And how do you handle the host cell protein spec in DS with regards to reagent change?
Well, so again, for host cell proteins, the specification is based on manufacturing capability. So first of all, it's important to do reactivity of your DS to make sure that you're reacting to all the host cell proteins that are in there. So typically that's done by AAE mass spec. And then also bridging. So if you have retained samples of your drug substance, you should test that in the current and the new assay to understand what impact the reagent change or the new assay is going to make, and then update your specifications accordingly.
And do you provide the specific host cell protein reactive protein list coming out of the AEMS analysis for specific kits?
So we do. So for whether you're doing coverage analysis for one of our generic kits, or if we do a process-specific kit for you, when we do the antibody affinity extraction with mass spec coverage, we generate a list of all the proteins that were identified in both the pre and the post, and that is supplied as part of the report.
Okay. Well, it looks like that's all the time we have for questions. So if we didn't get to your question, we will be passing those on to our speaker, and he can follow up with you directly. And there is still time to enter any last-minute questions, so go ahead and do so now. But thank you for joining us, and thank you to our speaker.
Thank you.
And the recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.